Hi guys, it's Mrs. Vance. I realize it's been a while since I made a video for you guys because normally I'm able to find videos by other people that are more authoritative on this than I am, but I've been looking for something that was really good and I haven't really found anything that I liked very well, so I'm going to take a stab at it myself. Anyway, we're going to talk about eukaryotic gene regulation. Last week we focused on prokaryotes and then moved a little bit to the stickleback since we had talked about that before, but talking about some of the things that are involved in eukaryotic gene regulation because it is a much more complex process than it is in prokaryotes. So, big thing is that there are many stages at which this can happen. Okay, there are in multicellular organisms, of course, it's very important for the gene expression to be regulated because the cells are specialized to do different jobs, and this regulation actually starts in the embryo because you know all those cells that start off in the embryo are just stem cells. Basically, they're just like a blank notebook that have to be primed with what they need to be done. And so gene regulation is how that takes place. So big thing is that once again, all of the cells have the same genome, but of course you don't want all of the genes expressed in every single kind of cell. And of course those can lead to imbalances and all kinds of diseases and things like that. And so there's a lot of expression, regulation of expression throughout the life of the organism, starting in the embryo. So big thing is there are lots of different things that can happen here. Of course, you've got signals that are gonna stimulate this whole thing to take place, and then chromatin modification can affect the expression of genes, the DNA, the unpacking of the DNA, making the gene available for transcription. It can be modified at the transcription stage, in the RNA processing stage by doing alternate RNA splicing. The transport of the RNA to the cytoplasm is another important stage at which things can be stopped or enhanced. And then in the translation stage, the RNA can be degraded before it's finished translating, or translation can be stopped in other stages as well. And then, of course, the processing of the protein can also be involved in this, as well as the degradation of the protein or the transport of the protein where it needs to go. So lots of different places that the regulation of the genes can be changed or can be modified or regulated in different ways. So there's a lot, it's a much more complex process, and frankly, I don't know as much about it as I'd like to know about it, and I don't think you have to know a whole lot of detail about it, but understanding some of the basics will help you overall. So big thing is that it's commonly controlled in all organisms at the transcription level, okay? And because eukaryotic cells are much more complex than prokaryotic cells and have a lot more things going on, there are lots of different opportunities for regulating the gene expression at different stages as well. So one of the first things we'll talk about is about regulation of chromatin structure. You know that during the normal everyday life of a cell, the chromatin it's kind of loosely packed in the nucleus. You don't really see the chromosomes, of course, because they're not tightly wound, but you still have a lot of organization going on inside the nucleus. The big thing is, you know, you've got the control elements of the gene, the promoters and activators and that kind of stuff. And so where those things are located relative to how the DNA is wound up around the histones and things like that can definitely have an influence on which gene gets transcribed when. So one of the big things that happens a lot of times is chemical modifications to the histones, and that affects how tightly or how loosely the DNA is wound around the histones. So two major ways that this is accomplished is by acetylation, adding of an acetyl group to some molecules. The positively charged lysines and histones is the main place for that to take place. And then that basically opens up or loosens the chromatin structure, makes them kind of unwind a little bit from the histone spools the DNA is wound around, and the addition of methyl groups can condense the chromatin and lead to reduced transcription. So one kind of opens it up and one kind of closes it down, depending on what's needed at the time. Here we have our compact DNA that is not accessible for transcription. So it's very tightly wound around the histones there, but when we add those acetyl groups, that causes the DNA to relax or in the histones to allow the DNA to unwind a little bit so that now the DNA is more accessible for transcription. Adding the methyl groups, of course, adds to different bases. That's usually to cytosine, and then basically they're in cells where the genes are not expressed, there's a lot of heavy methylation that goes on, and then oftentimes if you've got, like for instance, if you want to turn off a certain gene in a certain type of cell, it stays that way pretty much through every successive cell divisions once that cell is differentiated. There also can be histone methylation that can occur at other stages in life and actually can be passed on to the offspring. And so this diagram just kind of shows you a kind of a comparison of what happens with methylation and acetylation to tighten up the winding of the DNA or to loosen it up for making the genes more accessible. This is a little video from HHMI that shows some things about how one particular molecule can help silence the gene.
So here's the gene being transcribed, and next to it you'd see acetylated histones, nice open chromatin, and now you catch these little methyl cytosines. Methyl groups is appearing, and here comes the protein. It binds and will attract this protein complex along with the enzymes, the deacetylases, and now acetyl groups are gone and histones become tight, chromatin closes, no transcription factors can come through, and the gene is silent.
So just one example of kind of how that works in a cell. There are
other
things. I'm sorry. There are other things that other molecules besides that one. That's just one example of one molecule. There is some modification of the chromatin that, even though it doesn't alter the DNA sequence, it can be passed on to future generations. And I'm going to post another video for you that I showed to the kids on site last week. I've been showing the video for about 8 or 10 years now. It's about epigenetic inheritance. It's a SciShow video, so it's got Hank Green, and so it's entertaining as well as informative, and so I'll post that for you guys as well to watch that one. Anyway, big thing is that epigenetic modifications can be reversed, but not mutations, and so it's one way of regulating the gene expression from one generation to the next, basically. Okay. There are also chromatin-modifying enzymes that can basically make regions of DNA either more or less able to bind the transcription machinery, and that's not necessarily just about the histone modifications or the methylation or acetylation of the histone groups, but there are other things as well that can modify the enzymes. Most genes have multiple control elements, and these are basically, we used to say that a lot of the non-coding DNA was junk DNA, but we really are learning nowadays that it's actually more involved in the regulation and control of gene expression. And so there are various control elements and transcription factors that have to bind in certain places in a certain sequence to regulate gene expression in different kinds of cells. So that's kind of what we saw a little bit of with the stickleback activity. All right, this is about one thing that can happen, and that's RNA degradation.
The lifespan of mRNA molecules helps determine how much of a protein is made. Nucleotide sequences that affect mRNA longevity are often found in the 3' untranslated region following the stop codon. Enzymes attack the poly-A tail first, and eventually the rest of the mRNA. Depending on its ability to resist this degradation, a eukaryotic mRNA molecule may last from hours to weeks. The longer an mRNA lasts, the more protein is made.
So one more thing that can, again, if you don't want very much of the protein to be made, then you can basically turn on those enzymes that'll break down the RNA, or if you want it to be made many times, then you inhibit those proteins, those enzymes that can break it down. So here's a typical organization of a eukaryotic gene. Okay, we've got our enhancer, which is upstream of the gene itself. Notice it says distal. Distal means it's far away. It's not close by. There are also proximal or nearby control elements. The promoter is, of course, where the RNA polymerase is going to start, and then you've got the poly-A signal sequence there at the end that signals that this is the end of this gene. and then that's followed by the transcription termination region farther downstream. So here are proximal control elements. This is where we're going to start. We're going to start the transcription. Okay, here's our primary RNA, the transcript. This is called the pre-mRNA. In other words, it's what's been transcribed directly from the DNA. That, before it leaves the nucleus, has to be processed and the introns removed, the exons spliced back together, and then the messenger RNA gets a 5' cap of polyphosphate in guanine cap, and then a poly A tail, and the UTR means untranslated region. Okay, so here we've got the start and the stop codon, and there's our coding segment that's involved in our mature messenger RNA that will then be exported from the nucleus. Now, there are general transcription factors that act at the promoter, but then there are also other transcription factors that are going to that are often work with other control elements either nearby or far away from the promoter, depending on what particular cell it is and how those genes are being expressed. Here's another short video from HHMI that kind of explains some of the various elements that can be involved in regulating DNA transcription.
In eukaryotes, DNA transcription begins when general transcription factors bind to the promoter region of the gene. Those transcription factors facilitate the binding of the RNA polymerase enzyme to the start position of the gene. RNA polymerase needs to be activated by a type of transcription factor called activator. Some activators recognize and bind to the regulatory region of the DNA near the promoter region. Other activators bind to a distant region of the chromosome called the enhancer region. Activators bound to the enhancer region work with mediator molecules to initiate transcription. Another type of transcription factor, called repressor, inhibits the activation of transcription. Repressors can act in different ways. Some repressors bind to the binding site of activators and prevent activators from binding to DNA. Some interfere with the molecular interaction between the activators and the RNA polymerase. Some activators and repressors act by affecting chromatin structure. These repressors can alter the chromatin structure to make it coil up tightly. Coiled up genes are not available for transcription. Conversely, activators can uncoil a coiled section of DNA and make genes in those regions available for transcription. The interactions of all these factors determine the actual rate of DNA transcription.
So lots of different things involved in both nearby the gene, the proximal regions, and then also farther away, the distal regions. All right, so there are various transcription factors that was talked about in the video, okay? And some of them bind to the DNA, but some of them bind to proteins as well. And so basically the interaction of those with the RNA polymerase is going to lead to only a low rate of initiation rather than having all those enhancers and things like that. The proximal control elements are ones that are close by, like I said before. The distal ones are ones that are farther away. They're often called enhancers, and that's a lot of what you were showing in the Stickleback models that you were working on, is those various proximal and distal control factors as well as the mediators and things like that that are involved in the transcription of a gene. Activators bind to enhancers and stimulate the transcription. And they basically have two different parts, one that binds to the DNA and the second that activates the transcription, depending on whether it needs to interact directly with the polymerase or whether it interacts with a mediator. It just depends on the particular gene in the particular cell. Here's a another diagram that just kind of gives you an idea of how these interact with each other. You've got the activation domain there, and that's going to work with the part that binds to the DNA. The mediator proteins are going to involve various things about how those molecules interact with each other. There are a lot of different kinds of mediator proteins. It just depends on, again, it depends on the cell and the particular configuration of that particular gene. Let's see another little animation here about how the transcription is initiated.
Most of the fine-tuning of gene expression occurs at the transcription level, making an RNA copy of a DNA sequence. Recall that to initiate transcription, an RNA polymerase must bind to the promoter region adjacent to a gene. This cannot occur without the help of specific protein transcription factors. Some transcription factors, called activators, bind to enhancer regions some distance from the gene. The DNA bends, bringing the activators closer to the gene, where the activators interact with other transcription factors and RNA polymerase to initiate transcription.
So just another example of kind of how things fit together to work. So here we have some activators on this diagram. Okay, there's our promoter and our gene downstream from that. This is a distal control element. Okay, and so you have special proteins. Again, they're involved in every stage of that. There are special proteins that are used to bend the DNA. And then there are general transcription factors and our mediator proteins that are going to be necessary. And so all of those things interact and work together to stimulate the production of the RNA in the transcription initiation complex. So there are also repressors, like we've talked about before, with prokaryotes as well. And sometimes they can influence the chromatin structure, like it said in the video, or to either promote or to silence transcription. There are also a number of genes that have various combinations of control elements that can activate transcription when you've got certain ones present in certain cells. And this kind of goes back to what we saw with the Stickleback activity that we did. Okay, so this is DNA that's found in different kinds of cells. We've got an insulin gene, an albumin gene, which is a type of protein, and a crystalline gene, which is a gene to make the material that's in the lens of your eye. And so you've got enhancers for both of those things there that are distal to the control elements that are distal to the genes. Okay, in the liver cell, okay, you've got different activators available that are going to express the albumin gene, but not the crystalline gene. But in the lens of the eye, you have different activators available that will stimulate the production of the crystalline rather than the albumin. And so it just depends on various things. And again, it depends on the type of cell and what's going on there. Eukaryotic genes that are co-expressed like this are most of the time aren't organized into operons. There are some exceptions to that, but many times the genes are scattered over different chromosomes. But they have the same control elements, and so the activators recognize the specific ones, and then that allows those genes to be transcribed simultaneously and work together to do whatever the job is they have to do. Transcription is not the only thing that accounts for gene expression. Also, there are things that are involved in post-transcriptional modification of the regulation of the gene, and that includes RNA processing. There are various ways that the RNA can be processed and spliced depending on what is required at the time. Alternative splicing allows the genome to do various things with the same DNA sequences because you can remove different introns or put different exons back together. So here's another quick little animation about RNA processing, and we're just about done.
A cell can rapidly adjust to environmental changes by regulating several processes that happen after transcription. In RNA processing, a cap and tail are added, introns removed, and the remaining exons spliced together. Alternative splicing of RNA may create different mRNA molecules from the same primary RNA transcript.
And that just allows you to do different things with the same gene. So here's an example. Here's a gene. It's a gene for troponin T. The primary RNA transcript is shown here with the exons labeled 1, 2, 3, 4, 5. The introns are in between, and you can splice the RNA in different ways to make different combinations of those introns to make different protein products that are necessary for different kind of jobs. Also, you can initiate translation of those or block the translation of messenger RNAs with various kinds of regulatory proteins that can stop the translation or enhance the translation. And so it just depends on, again, there's lots of different things that happen. There are certain ones, translation initiation factors, that are activated in an egg following fertilization because there's a lot of stuff that has to happen really quickly all at the same time. mRNA molecules can last different amounts of time, and so part of that has to do with how much of the protein is needed at that particular time. And there are various nucleotide sequences that influence this lifespan, and that's in that UTR, the untranslated region at the 3' end there. And then after translation, there are various kinds of protein processing things that can happen in terms of rearranging, chemically modifying the polypeptide, cutting parts of it and putting different parts on there. It just depends on how, on the needs of the cell at that particular time. This has been a longer video than I really intended it to be. I'm sorry. I'm going to stop talking now, and I'll make another video for the next part.